So let's have a look at how we can analyse molecular sequences to infer evolutionary relationships. The syllabus objective says you need to be able to analyse data from molecular sequences to infer species evolutionary relatedness. So we need to assume that the differences in molecular sequences can be used to determine evolutionary relationships, and that organisms with similar sequences are more closely related. So here's a very simple example here of some molecular sequences from 4 different organisms. They've been aligned so we can compare them directly. Obviously, this is only 18 nucleotides, where normally you probably have thousands of nucleotides in which you can compare. These are DNA nucleotides we're looking at here. And we want to compare it to A, species A. And so that's why I've said it's 100%. Species A is 100% exactly the same as species A. Now we're going to compare that to species B, compare species B to species A, etc. So these are the different nucleotides, and then you can probably have a look down here and see that there's some differences with B compared with A. So for example, in the first 3, we see we've got an A instead of a G. And then we've got a T instead of an A, and we've got a G instead of a C. So to work that out, there's 3 differences or 3 mutations here compared with A. So that means that B has 83% similarity to A. Now you can go ahead and do the same thing for C and D if you like. With C, there's only 1 difference, so that gives us 94. So the way I did that is I just worked out, okay, so we know there's 18 nucleotides here for, in total, and one of them is different. So 17 nucleotides are the same for C divided by 18, times by 100, that gives us 94%. So if you want to try and do that with D, you'll see that there's a couple of differences. There's 1, 2, 3, 4, 5 differences. So that would be 13 divided by 18. So 13 of the similar. 13 divided by 18 times by 100. That gives us 72%. I've just rounded them. Okay, so that gives us an idea of the similarity between these different organisms. And from that, we can work out the evolutionary relationship and perhaps put it onto a cladogram. Can you work out what would be the organism that would be closest to A on the cladogram, and which one would be furthest away? D would be furthest away because it has the least similarity, and the next one, of course, would be B, and then the next one, C. So here's a question from a previous external exam. We've run through this one before. We can go through it again. Firstly, you need to determine whether this sequence is correct. So press pause and see if you agree that this is the right sequence. Now the next thing you need to do, I agree that this is actually the right sequence based on these similarities and differences. So in other words, D is most similar to A, and B is least similar to A, and that's why they're in that order. So determine whether it's been drawn correctly, and then explain your reasoning. So your reasoning would be then talking about referring to this data to explain that, say, B is the least similar, and you could actually work out all of those differences and make that as okay, so B is the least similar and D is the most similar. So that's all we really need to do to be able to analyse molecular sequences to infer evolutionary relationships.